hopefully this works. I hope it does. We'll see when we get the results. Oh, that is not the chapter we're on yet, though. Okay, so where we were last time, we've been looking at the cell, the components of a eukaryotic cell. Last time we met, we did together, we kind of finished this drawing together. And as I was mentioning to other people earlier, the ability to draw something meaningful for yourself. Maybe it's a figure from the book that helps explain something to you. Or maybe it's something like this, where I've kind of cooked this up, which then goes into a lot of the details we did in class. To be able to draw it for yourself from memory, that's a very powerful way to study. As a matter of fact, what you could do is you could do this drawing, but maybe on a legal sheet, so even longer, right? Longer piece of paper. And out to the side, you could add notes about each of these topics. So essentially, you're recreating your notes in a pictorial format. I know a lot of you learn better that way. And for sure, what you do is, if you were to do that, and you were to make a drawing like that with notes added in on the sides, and then if you were to redo that drawing, draw it again for yourself, and then maybe get a blank sheet of paper out, put everything else away, and try to do it again from memory. If you do that a few times, you get better every time you do it, and you learn it, and it doesn't give you a headache. It's a great way to study, because it's kind of like a competition with yourself to try to do better each time, try to remember more each time. Fantastic way to study. Now that we're into this kind of stuff, where it is kind of largely a lot of pictures and concepts that can be represented pictorially, that can be really helpful. But anyway, that's what we did last time, kind of getting into the components of a cell. Now, we're getting to components of a cell now that are not as easy necessarily to represent in a picture of that drawing, but these are these protein-based fibers that give the cell internal support structure, what we call the cytoskeleton. So we got that to go, the cytoskeleton, as well as then the stuff that is on the outside of the cell or that joins two cells together. So that'll be the last topic in this chapter, and then we'll be ready to move to the next chapter. But for now, where we had gotten to, we did, did the structures that help us store and utilize genetic information, the nucleus and ribosomes. We did the endomembrane system with the ER, transport vesicles, and the Golgi apparatus and lysosomes, and eventually the vacuole. And then we did energy transforming organelles like mitochondria and the chloroplast. Okay. And so now again to the cytoskeleton. What we say about the cytoskeleton, it's there for support, it's there to give the cell its internal structure and shape, but it's also for moving stuff around. Especially two of the fibers of the cytoskeleton are not just there for support, but they're also for helping to either move the cell places, or maybe change the cell shape, we'll see that in a minute, or to, very importantly, so that stuff in the cell can move around with a purpose, with a direction, not just floating around free arbitrarily, but moving from point A to point B in a relatively straight line. And that's kind of things, those kind of jobs are what are going to be great about the cytoskeleton. 3 different kinds of fibers for the cytoskeleton: microfilaments, intermediate filaments, and microtubules. Of those, we're going to see the first and last are really kind of the more important ones. We'll get to those in a second. Okay. So here's a figure from the book that just shows the 3 different kinds of cytoskeletal fibers. You can pay attention to that if you'd like. What's more important for me is that we get these in the notes. All right, so we should be picking up a new material here. Is that right? Okay, good. Not much to write on this slide for those of you who are writing with the fill-in-the-blank notes. Just the word actin. Actin is the protein that the microfilaments are made up of. So microfilaments are long. You know what a filament is? Or when I say something is filamentous, what does that mean?
Yeah. I thought of it as like these long lines.
Mm-hmm. Yeah, like a long lines. Mm-hmm. Good. Kind of rod shaped, okay, or like sometimes we think of a filament as like a strand, but its shape is not like round and blobby. It's kind of a straight line, or it can be a straight line. Okay, that's when we say filament. That's what we're talking about. A microfilament are pretty small, as the name implies, but they're made up of, and get this is kind of blowing your mind, right? We learned how polypeptides are polymers of what? Do you remember?
Proteins.
Well, they're proteins, but what are the monomers that make up? Amino acids, right? Very good. Very good. See? Now we're going to see that we can take proteins themselves and link them together, not like covalently bound together so that they're permanently attached, but non-covalently but still pretty strong. And that then can form a different kind of a polymer. We can actually make a big long chain out of proteins. Do you get what I'm saying? So the protein itself is a polymer of amino acids. But now we're going to take those proteins and link them together in a long chain, and that's a different kind of a polymer. And so that's what these microfilaments are big long chains where each unit is itself a polypeptide. The polypeptide units are called actin. Technically, they're called G-actin, stands for globular, because if you looked at any one of these proteins, it does kind of look like a blob, doesn't it? You see that there? It's kind of round, not round maybe, but kind of boxy in shape. Okay, it's definitely, itself is not a long fiber, but when you link a bunch of them together, then you get a fiber. Matter of fact, you take two chains of them that kind of spiral around each other. Do you see in the picture? Okay, and that's the what we call F-actin, or the microfilament. Okay. So microfilaments are made of actin. You get two chains linked together, twisting chains. I already said that. Right. Okay, in the fact that a filament, absolutely, yeah, you can think of it like that. Now, what will happen in your cells is that DNA itself wraps around protein, so its actual shape, only through short stretches would it actually be kind of like a straight line. Otherwise, it would kind of be wrapped around something.
But it's sort of like the same concept.
Definitely, yeah. If you had naked DNA, you could think of it as a filament, absolutely. Yeah, very good. But different in that here we're making a polymer out of polymers, right?
Yeah,
each protein subunit is itself a polymer of amino acids. Okay. All right, good. So those are microfilaments. That's their structure are made up of two chains of actin twisting around each other. Now, as far as what they do, besides forming structure, is that they help things move. Oh, I guess that's not the point I'm at yet. Sorry. It says flexible and dynamic. What does flexible mean? Well, that's pretty easy, right? Right? It can bend. It bends, doesn't it? So a microfilament can bend somewhat, okay? Bend but not break, right? So that's what microfilaments are good for. What does dynamic mean?
Intense.
Active, intense. I can see that, but that we often I think sometimes in the vernacular we would conflate those two things, but I think more like active, alive, changing. Dynamic means changing, or at least changeable. So remember how I told you that actin is made of a polymer. It's kind of a polymer of polymers. What that means is, and by the way, these polymers are not bound together covalently and like super strongly. They're bound together what's called non-covalently. That means that you can put a big bunch of them together in a chain, but then you can also break that chain apart. You can dissolve. And you can take those monomers that are those little protein subunits and then go build a new chain of them somewhere else. So that's what I mean when I say dynamic. It means that they can be changed. That just because you've built a microfilament doesn't mean it's going to last forever. It can be broken apart and reassembled somewhere else to do a different job. And that's really important when you consider the kinds of things that it does. For example, it allows movement of a cell. Hopefully tomorrow we'll see amoebae in the lab. I bought some amoeba to look at under the microscopes tomorrow, and you should look at it after the lab quiz. By the way, advertisement, lab quiz tomorrow. That means open lab today from 3 to 5, but also open lab tomorrow from about 8 a.m. in the morning till about 11. The lab will be open, and I'll be there to answer questions. Okay. About 8 to 11. After that, I'm going to need to eat lunch and get ready for lab and set up the lab quiz.
today 3 to 5.
Today 3 to 5, tomorrow 8 to 11. Okay. Also remember to look at the study materials from that list. All right. End of advertisement. So the thing about actin filaments or microfilaments is that things can move on them, and there's this protein called myosin. It's a motor protein. Motor protein means that it can help things move. So, for example, that allows the amoeba to put out what's called a pseudopod or a false foot, and then kind of extend part of its cell outward, and then grip onto something and move the whole rest of the cell behind it. That's how amoebas move, is by that pseudopodial movement. Also, for that matter, myosin works in, you probably know this from anatomy and physiology. It's fascinating. You guys will learn this if you haven't already. When you get to general biology 2, you'll learn how muscle contractions work. That's one of the coolest things I've ever learned about biology. But the short version of the story is you've got microfilaments over here, and this protein's called myosin over here, and they sort of overlap. And the myosin can use some ATP and do a motion that causes the fibers to slide across each other like this. So you went from just barely overlapping to like this. And what does that cause if you're talking about a muscle? That's actually what happens during the muscle contraction. That movement amplified over thousands is too small a word. Many of these little units called sarcomeres where that happens. It's amazing. I don't have time to tell you the whole story now. You'll learn about it in general biology 2 or if you take physiology. Yeah.
Okay, so this is kind of what I'm understanding about this slide, but you're saying that there's actin, like in the cells, cytoplasm or something, and then there's other parts of the cell that have myosin, and then in order for that part of the cell to move around the cell, you need to work with actin to move, because they're dynamic?
Well, the dynamic bit means that you can build actin chain and then break it down and build it again somewhere else. Okay. Now, so that's not when I said dynamic, I wasn't talking about the movement aspect of it, although that's really important.
Yeah.
Okay? So the myosin aspect of it is you have, myosin can be attached to something solid on the cell, like, I don't know, maybe an intermediate filament, or we'll talk about that in a minute, something else that's giving it kind of a plate, a place to grab onto the cell. And then it can grab onto the actin and do a motion, and by doing that, you could kind of pull the cell along where it can cause muscle contractions.
Yeah. Okay.
So myosin is the motor protein, actin is what it grabs onto and does its motion. And the motion does look like this. So if this was actin and this is myosin, then the actin grabs onto it, flicks like that. And that's actually that motion of the muscle contraction. Yeah.
So is myosin only found in muscle cells
Oh, no.
and amyloid?
No, I would not think so. Any cell that, that's a good question. We're getting beyond where I feel comfortable answering that question.
Okay.
But I don't think so. I think there are other cells that would have myosin as well. Certainly, right, because, of course, actin myosin is an animal cell in the process of cell division. It's the process of pinching two cells apart from each other. That's controlled by myosin and actin. So yeah, every cell is going to have, every animal cell is going to have it. It needs to do that cell division process.
Right.
So let's not get bogged down onto it. So we said muscle contractions, amoeboid movements, and then in plants there's this thing called cytoplasmic streaming, which is also apparently driven by actin and myosin.
The only reason why I asked that.
We'll see cytoplasmic streaming tomorrow in the lab when we look at plant cells.
Yeah. Okay. I just wanted to ask that because the only time that I've ever really heard of actin and myosin is in the muscle.
But there's other. Okay. The second type of cytoskeletal element is intermediate filaments. They're kind of boring. You write those things down, and we'll talk about them briefly, but I'm going to skip past intermediate filaments. Yeah, this is a different kind of keratin, but like more dense. I think keratin kind of has a couple different versions or a couple different forms, but yeah, it's the same kind of stuff. So she was asking about fingernails and hair being made of keratin, yeah. But within a cell, you have keratin, this intermediate filaments, because they're not broken down easily, remember, they're not dynamic. What's the opposite of dynamic? Static, exactly. That's static. Once you make it, it's there. Okay, it's going to stay there. If you notice kind of the shape of what's going on here, does anybody recognize what kind of cells these are? They have microvilli. Lipid is a type of macromolecule. What kind of cell are these? That have, yeah, you know, you've had anatomy and physiology. So these would be cells of your intestinal lining. So there are on those cells to increase surface area, right? Because you got all these waves, right? And when I tell you, anytime you see bends and curves and waves and stuff like that on a membrane, it's to increase surface area. So you got these little projections that stick out of the surface of the cell, out into the gut lumen, into the middle of your intestines, and that's where you're absorbing your nutrients across into your body. So if you notice, the structure of these are supported by microfilaments. But underneath that, once you get down into the heart of the cell, then the cytoskeleton kind of changes and is more based on intermediate filaments. Another thing is that in your skin cells, these cells here are the cells at the bottom of your epidermis. So these cells that I'm running across right here is the epidermis. Down here is the dermis. So when you take it, we'll actually probably see that tomorrow in lab. Anyway, so what happens is the cells here at the bottom layer of the epidermis, they're alive and dividing. But as you move out from here, as they divide, then those cells kind of are shoved outward. And as they move outward, the cells die. And out here, they're dead. And basically, all that's left of them is the keratin, pretty much, is the keratin, because that's so tough and it doesn't degrade, it doesn't remodel or anything like that. So what's basically left out, they're called keratinized cells because they're dead, and pretty much most of what's left is the keratin. Okay. All right. But otherwise, it's kind of boring because stuff doesn't move around on them, unlike microfilaments and the next one, microtubules. Okay. And I think of microtubules as a molecular biologist. I think of microtubules as the most important. So tubulin, which is what it's made of, there's two different kinds, and they dimerize, and then those start to form these little, what's called a protofilament. But don't worry about that. Look, here's the thing is that they organize into this shape that kind of spirals around, as you see, and the effect of it is, when you get a bunch of them together, is you get a hollow tube, kind of like a drinking straw. But as far as I'm aware, I don't know of anything that actually moves in the middle of this drinking straw. I mean, sure, there would be water molecules in there, but I don't know of any important thing that actually moves in the middle. What is more important is the stuff that can attach on here to the surface, to the outer surface, and move around, because this molecule is, the microtubules are very important for moving stuff around within a cell. This picture is so lame. It's really kind of pixelated. I don't know what happened here. So microtubules are dynamic. You can, if I didn't say that anywhere else, they can be built and unbuilt and rebuilt, unlike intermediate filaments. And most importantly, there are what is called motor proteins that move things around within the cell. For example, we learned about how a vesicle might bud off of the endoplazmic reticulum and move to the Golgi apparatus. And then eventually over here on this transface, a vesicle bloods off the transface and moves toward, for example, the plasma membrane. Well, how did it make that motion? How did it know where to go? It's moving along microtubules. They act like train tracks for the cell. Things are transported around. Yes?
But you said that they could be broken apart?
They could, yeah. They could be, those microtubules can be depolymerized. In other words, broken down back into their tubulin monomers, yeah.
So is there a set amount in one cell?
It's a great question. I think it would just depend on what the cell needs. Some cells would need more of them than others. And certain times of the cell's life, it would need more than others.
Well, that's why I'm asking, because, I mean, if it has a set amount, then only so many can go in that one spot one time, depending on what's needed.
So, here's what I need you to eventually get, whether you get it now or not. Cells are alive. If they need some more of something, they can make more. See, because in the nucleus, there's a gene for alpha tubulin and there's a gene for beta tubulin. And if you need more of that stuff, you would just, remember the central dogma of molecular biology? You just transcribe more of the messenger RNA for that gene, and then the messenger RNA goes and finds a ribosome and gets translated, and so you make more of the protein. That makes sense. And then if you've got too much of a protein, it can be degraded. We haven't talked about that yet. Okay, awesome. So, the motor proteins are called kinesin and dynein, and the reason we have two of them is one of them, there's actually a polarity to this molecule. There's a plus end and a minus end. You don't need to know this for the exam. But one of them, motor proteins, moves from plus to minus in its direction, and the other motor protein moves from minus to plus. So you need two different motor proteins because there's two different ways that you travel along. Make sense? Good. And so these are really critically important for moving. Okay, a couple more things about microtubules. Move stuff around in the cell, including moving your chromosomes when you're doing a cell division. Okay, so there are these little beasties within the cell called the centrosome together, each made up of two centrioles, and these guys act like Grand Central Station. Remember I said the microtubules like train tracks? If that's true, then these guys are like Grand Central Station. They kind of organize and kind of are the hub for the microtubules kind of emanating out of there. And that's especially true when it comes time to separate your chromosomes in cell division. Hang on to that because later in the semester we'll talk a lot more about cell divisions, mitosis and meiosis. Rebecca, you're just laughing because you're like, yeah, I already know this stuff. You must have had a pretty good biology class somewhere. Okay. Oh, except that I already asked you to define centrosomes. Who's got a definition? What did your definition say? It's funny because I forgot that was there, obviously, or I wouldn't have defined it for you. Okay. Huh? Yeah, Sana. Tubule. Good. Yeah. It's kind of like so it's kind of the hub for the microtubules. They all kind of connect back there. You can think of that as the case. Great. Do you understand what I mean when I say Grand Central Station? No? So with trains, a lot of times what you're going to find are these hubs where a lot of train tracks kind of coalesce and come together. And like say if you're a passenger on imagine you're a passenger because that's kind of easier for you to imagine than being a cargo train car. But if you're a passenger, then in a passenger train, then you might go from one city, or like a small city, to a hub, a city that's nearby. Okay, and that's where a bunch of trains all come together. And then, like they're coming together, a bunch of trains also leave out for various destinations. And so, you know how it is when you fly. Like when you fly on everybody's probably familiar with this, right? If you've ever flown anywhere, if we were to fly out of Reading, you're not going to fly directly from Reading to like Orlando, Florida, or something like that. No, you're going to fly from Reading to San Francisco. San Francisco's the hub. Okay. In the same way with train tracks, now we're going to bring our analogy back to train tracks, they're, from a smaller place or a smaller station, the train would go kind of first to a hub, and then from there you can pick any number of directions you want to go because there's a lot of directions leading out from that hub. In the same way, the centrosomes are like the hub for all the microtubules kind of emanating forth out of it and going all over the cell from there. All right. I'm going to reveal these things. All right. I don't think I need to say anything more about that right now.
Why don't plant cells have them?
It's a great question. I don't know. And I'm a plant biologist, so I don't know. Maybe they don't have them like obviously visible centrosomes. But they have microtubules. It's very important. Yeah, it was just maybe organized a little differently in plant cells. I don't know. Good question. All right.
So are they organized differently?
I do not feel comfortable answering that question. I would need to look more into it. It's not something I ever think about. So. I try not to tell you things that I'm not sure of.
Which is smart.
I always feel real, because I'm not a physicist, when we get into the parts of this class that deal with physics, I always have to check what I'm saying with actual physicists and say, okay, am I saying heresy here, or is this about right? And I did check it with my physicist friends, and they said, yeah, you're good. Okay. Another thing about microtubules is that we use them, we use them to move around fast. In a way that there are these things called locomotor appendages. Now, what's an appendage?
Something that is stuck on, or is it jerked out?
Right, it comes out of something. Like you got a main body, and like my arm is an appendage to my body, right? Okay, so it's not part of the main core, but it kind of sticks out, right? So an appendage like this is like a part of a cell that kind of sticks out. Okay, there's two major kinds of locomotor. Now, what would locomotion be? You guys know this, right? Moving, right? So these are appendages for the purposes of moving. Like my legs are my locomotor appendages, pretty primarily, right? You know this, right? Okay, so there's two major kinds of locomotor appendages that cells use at that level, and they both use microtubules to support and move them. And the analogy here is to how swimmers move. And I'm a swimmer, some coach, so let's see if this will play. Oh, darn it, it's not there. Okay, well, that means I'm just going to have to demonstrate it for you. Okay. So this is best demonstrated by the butterfly stroke. Okay. Actually, I'll show you in a second. Let me give you notes first, and then no, I don't want to do. I didn't mean to do that. Okay. So you got flagella and cilia. What's a flagellum? I'll show you the butterfly in just a minute. Yeah.
It's a tail.
Okay. It's a tail. Okay.
I can elaborate.
Okay. Can you elaborate? Okay.
Yeah.
So how does it move?
It's one appendage.
Usually there's only one or two in a cell.
Yeah, on the back of the cell that rotates counterclockwise or clockwise or wherever it's going to go.
The effect of that is that it
Moves forward.
kind of like a whip-like motion
Yeah.
that drives the cell forward away from wherever that gel is pointing usually.
Whip-like motion.
Whip-like motion, that's correct. Okay. Good. And what about a cilia? A cilium, technically. Cilia would be plural. So I think that's right, cilium. There's also cilium that's like a nutritional thing, but that's spelled differently. Cilium, I suppose here would spell like that would be
Same thing, but they're
smaller.
A
A lot more of them, but they move differently.
Yeah.
Right. Rather than being a whip-like motion that drives forward, they have two parts of their motion. They have what's called the power stroke and the recovery stroke. So this is best demonstrated by the butterfly. So get ready. This is my embarrassing attempt to do a dry land demonstration of the butterfly. Have fun with this. Don't videotape it. Okay. So on the butterfly stroke, there's the legs and the arms. So the legs are kicking like this. It's called the dolphin kick. Which one is outside of the water. Does that make sense?
Yeah.
So that's like the cilium. There's only two. Of course, in the cilium, you would, real organism, there's many, thousands of cilia. Okay. We'll see some cells tomorrow that have cilia, okay, and that's how they move. We'll probably also see some cells tomorrow that move by flagellar motions. If you have flagellar motions, the cell tends to kind of wobble around as it moves. It's kind of because it's got one thing that's moving like that. Whereas cilia movement is smooth. It's like butter. It's really drifting, really nice.
So we have like tomater versus.
Yeah, something like that. Okay. Yeah, that's true because those ciliates, man, they really cook. They go through the water fast. Okay. Oh, I forgot that this was here. But it's good. It's good to know. We're transitioning from one type of organ to another. While we're here, might as well take advantage to say, coming up before too awful long, we'll have what's called the cell membrane drawing too. That it may make more sense for you to wait a little while because the next chapter will be all about cellular membranes. So you get a sense of what that's like. So cellular membrane will be drawing basically the foundation of it will be that phospholipid bilayer, which you're also going to put other stuff in it, as will be indicated by the figure that I refer you to look at, and also some stuff I put online, some pictures that can help you know what to draw. And so ask me any questions you got about that, and then eventually later to the end of this month or end of next month, it's still September, I guess, for one more day. Toward the end of next month, we got bioliteracy number 1. Not bioliteracy. This should be This may be biology through the eyes of faith, but I need to check that, so I don't. That may be an old announcement that doesn't mean anything. Sorry. We used to have bioliteracy in this bioliteracy assignments in this class, but I gave them up. Okay, moving on. Bioliteracy basically was when we would look at like scientific papers and try to start reading those. Eventually, I moved to where we do that more in lab. 1023. So sorry about that. All right. So that is biology through the eyes of faith. Okay. All right. So I was going to do one more thing before we left the idea of the cytoskeleton completely. If you like a table, we can do that. We've got microfilaments. No, sorry. We've got intermediate filaments. And we've got microtubules. If you like things organized in tables, this might help. This is basically, I'm not giving you any new information here. It's just stuff that you already knew. Sorry. There we go. I don't know what happened there. Okay, yeah. So, over first one, we could say made of. Made of what? Microfilaments? Actin. Mm-hmm. Okay. Intermediate filaments made of keratin. Okay. Microtubules. Tubulin. Very good. We'll get there in a minute. Okay. Next one. Dynamic or static? Okay, so microfilaments are dynamic. I'm just going to give it a D. Okay, intermediate filaments, static. Microtubules, dynamic. Okay. Oops, probably shouldn't have done that. Well, this is fine. Let's see. Movement? Question mark? Microfilaments? Stuff move on it or with it? Yes. What I meant to do here, let's change that. Let's just skip that one and we'll go motor proteins. How about that? Okay, that's better. Okay, here it's myosin. Okay, intermediate filaments. Not applicable. Okay, and microtubules. Kinesin and dynein. Okay, and if you wanted to fill out the rest of that by giving examples of the kind of movement that, which of course would be not applicable here, you could look at muscle contractions. If if you want examples of the kind of movement, muscle contractions, amoeboid movement, versus moving stuff in the cell and locomotor, like cilia and flagella here, if you wanted to fill out the rest of that table. I'm not going to do that, but if you like the table, there you go. That might help. Although I found that, well, it helps some of you. Oh, did you do this one? It's not written, it's kind of sloppy, but I was hoping you'd pay attention as I was writing along. Keep right with me. Okay. All right. Okay, so far we've done things that exist inside of here, inside the cell. Now we're going to actually start looking at some things that are outside the cell and some things that exist that hold cells together, two adjacent cells together. So that's this next kind of component. So some terms or ideas. The extracellular matrix is that stuff outside the cell. And I'll let you just write all this together in the connections. I found a long time ago from my guests that if I try to talk and explain things while they're writing, it actually does no good to anybody. So I try to, if they're taking notes in class live, then I try to give them a chance to write it down, and then we'll explain it to them. It slows things down, but at least it works. I'm trying to think when I can go.
I've got some things that I usually use as an example here. Let me run down to the lab and get it real quick. We can finish writing that. Off you are.
I have the key.
Years ago, I found these floppy disks in the faculty staff, faculty break room or something, and I thought to myself, these things aren't good for anything anymore, except I can glue them together and actually use them as models of cells. I can make little boxes out of them that I can use as models of cells to explain this very concept that we're about to get to. So hold off on that, and I'll get to it. Okay. So we got extracellular matrix and connections. Let's start with extracellular matrix. Extracellular matrix. We'll start actually with how this plays itself out in plants. Yeah, I'm still catching my breath a little bit. When I go up the stairs, I take them two at a time, and okay, so we're talking about, let's look at the plant first of all. If we look at a plant cell wall, or plant extracellular matrix of cell wall, usually you got a couple of different parts to that. You got the primary versus secondary cell wall. I'll let you write that down, and I'll draw them a little bit for you. Should have changed these slides and did that right before I left. That way you can have been writing, using that time productively while I was going to grab my boxes. Let's see, do I have one more? Nope, sorry. Humans, no, we don't have cell walls at all. Okay, cell walls is a plant thing, it's a fungi thing. Bacteria have cell walls. Our cells do not have rigid, polysaccharide-based structures out there at all. Our cells do have extracellular matrices, but they're not as rigid as plant cell walls, and they're based on protein. Well, glycoproteins, maybe a little bit what we call proteoglycans, something like that. Do you have a question? Okay. So the extracellular matrix of a plant cell, like so here we've got two adjacent cells, and they're connected, you'll see in just a second, by a connection that we'll talk about when we get there. But first of all, what's in between them? Well, there's this bit in the very middle. There's this glue-like substance called middle lamella, which is protein-based, and it kind of acts like a glue. So then, outside of that, and I'm just doing a cross section here. So this little hole, this passageway between the cells that I've drawn, would be kind of just like a small little circle, like a small tubular passageway. It doesn't run all the way through the cell wall. It's just like, or it goes all the way from one cell to the other, but it's otherwise just very cylindrical, and it doesn't take up, so it doesn't like go across the whole axis of the cell, the space between the cells. So just on either side of the middle lamella, there'll be what's called primary cell wall, and that's the cell wall material that the plant cells secreted first. So I'll do that in blue. Maybe I should have done a different color because blue and black can be kind of hard to tell apart sometimes. At least you're getting color this year. I looked at last year's drawing, it didn't have different colors at all, so what are you going to do? So these are the primary cell walls. Again, that was actually secreted or made by these plant cells first, and then after that, they put down a layer of secondary cell wall. And you can tell that it was after because what they did after pushed the primary that they made first out of the way, right? So that's the reason why the secondary cell wall is actually closer to the cell that has made it than the primary. Does that make sense to you? That if you made this first and then you made this second, what you made second would push the other one further out of the way. Okay, so the secondary cell wall then would be in here. Not all plant cells have secondary cell walls. Only, like majorly, you'd have like the ones that are really tough, like the fibers that make up the vascular system, phloem and xylem and stuff like that. They're going to have secondary cell walls because they need to be rigid and long and tough. And they actually, those cells are actually the ones in a tree that makes up like the heart of the wood, right? Like the real solid structure. Yeah.
So, are the cell walls durable enough to sustain itself during the lifetime of the plant cell? Or?
Yes. Yeah, yeah, that's right. Once they're made, they're not going to be remade. No. No, that's right. Now, certainly by the time a cell puts down secondary cell wall and it's done with that, that's going to be forever. Or I say forever, until it's burned, right? Because like this is what we do when we go out in the woods and have a campfire. Burn the cell wall. Okay. Anyway. All right, so those are the layers of the cell wall. I also talked about middle lamella, which is in between them. You should probably also label secondary cell wall, right? Second. Okay. The other thing about plant cells is they have connections that join adjacent cells. And these are called plasmodesma, singular, or plasmodesmata in the plural. You'll see some of these tomorrow. They're very difficult to see. They're so small that they just kind of look like hairline cracks in the cell wall material. You have to kind of really look carefully. You're probably aware, right, that you, as a person, started out your life as a single cell, right? We call it a zygote. And then the cell divided a lot and grew in between the divisions. Same thing with a plant. Each plant started out as a single fertilized egg, which then divided a lot. And so what that means is that if you're looking at with two adjacent cells within the plant, these two cells were probably at one time the same cell. They probably are descended from the same cell. But as that cell divided, and then started laying down laying down wall material in between, they didn't quite cut each other completely off. And so they left this little tunnel between the two cells so that material could flow back and forth between. Water can flow through this, also proteins and some signaling components of like the plant uses molecules to signal different things to itself. That can flow back and forth across there through the plasmodesmata. Okay, we're good so far. Plasmodesmata, a lot of people mix that up with a similar one from animals, which is what we're going to get to now. Animals also have cellular connections. There are 3 different kinds of cellular connections in animal cells. But we'll get to the extracellular matrix first. This, by the way, is very close to the kind of drawing you'll be doing with that cell membrane drawing. Okay, something that looks a lot like this, maybe with a few more things going on. Maybe, there's a whole lot going on here, but maybe you don't have to draw as much extracellular matrix or as much cytoskeleton, but you're definitely going to want to do more things going on across the membrane because I think I asked for 8 different components that will be in there other than phospholipid. Anyway, so here we have a protein that's embedded in the membrane that connects the cytoskeleton here to the membrane, also can connect the extracellular matrix to the membrane. Notice that there's these little green-looking branches. That's the way your book is depicting what's called glycoproteins. So these are sugar chains that come off of the proteins. Okay. All right. Any questions so far? Anyway, this is stuff that's outside of the cell. We consider this to be the outside of the cell, but it's what's called the extracellular matrix. Okay. In animals, let's write this. Let's see. So if you have 2 cells that need to stick together, I think of like we were looking earlier at the cells of the gut endothelium, gut epithelium, the lining of the gut. Those cells need to stick together pretty tightly so that stuff doesn't leak, so that like material from the inside of your digestive tract doesn't leak into your body. You want to tightly control what goes across there because there's bacteria in the gut lining, there's food that you may not want to absorb. There's stuff, a lot of stuff that you don't necessarily want. Matter of fact, there's a word for it. You probably heard of this. If somebody has a leaky gut, you ever heard of this idea? You don't want it, okay? You want your gut to be nice and tightly, the cells to be tightly tied together so that you can tightly regulate what goes across there into your body from your digestive system. So we want to be able to glue cells together. Also, some cells need to grab onto another cell so it can move. Cells in your body. Throughout your development, you had cells that were formed in one part of your developing embryo that rely on the ability to migrate in different parts of the body and set up shop somewhere else. And those cells have become important parts of your body, but they were formed in a part of the embryo that was way far away from where they eventually end up. So that's cell migration, and then it allows cells to communicate together. together. So this is extracellular matrices out there. Then I want to talk about the connections among animal cells, and that's why I went down and grabbed my boxes. There are 3 different kinds of connections in animal cells. The first one are called tight junctions. This is the one, if you notice, prevent fluid from leaking, right? That's what I was just talking about. A tight junction would be comparable. Let's imagine these 2 boxes represent cells, and you can kind of see into them. We cut the tops off those cells, but they're adjacent cells, they're right next to each other. A tight junction would be like maybe if I took some super glue and laid a bead of super glue along through here and then press them together. Although I guess super glue isn't easy to dissolve. Maybe instead we can think of it more like, if you can imagine if I was able to do this, these are made of hard plastic, but if you can imagine them being made of something softer, imagine if I was able to take a needle and thread and sew back and forth in a line. And so you don't have a connection just at one point, but you kind of got this whole kind of axis across which they're connected, not just at one point, but across the little ways, right? Now, could you imagine that could prevent fluid from being able to leak in between these 2 cells? Okay, so a tight junction is kind of like super glue laid across as a bead, a little line of super glue between the cells and pressed together, or like sewing them back and forth along a line. That's what tight junctions are like. Or like Ziploc seals can work as well. All right, we're good so far? Okay. Second type of connections are desmosomes. Desmosomes would be like if I drilled a hole, kind of a decent sized little hole through here, and then put a nut through on one side and attach a bolt to it on the other side. So now I've got a really firm connection, but just at one spot. Does that make sense? As opposed to the tight junction, which was kind of across a line or an axis, the desmosome is just a really firm, but at one spot only.
So if you have that and the cells start to move away,
That's
what
going to
happen?
Well, if it would resist them pulling away from each other to a point, but at some point you're going to break your cell membranes, right? So it depends on how hard they're being pulled. Right?
Why would they be pulled that hard?
Muscle strain, or any force. I mean, there's forces. You subject your body to forces all the time. For example, I could hit that real hard. Well, that is putting a lot of different kind of forces on that skin. And it could be damaging. It would, they'll be able to resist a certain amount of force, but at some point, everything's going to break down. All right. And then the last type of connection would be a gap junction. One of the most common mistakes I get is that students confuse gap junction and plasmodesmata, and almost rightfully so, because they're very similar, right? Plasmodesmata are connections that allow fluid to pass between adjacent plant cells. Here, gap junctions perform kind of the same role, but with adjacent animal cells instead. And so the difference is animal cells don't have that cell wall that they have to go through, the tube between the 2 cells has to go through. So a gap junction would be like, here's my 2 adjacent cells. Let's say I drilled a hole in all the way through, and I didn't put a bolt in there. It's just an opening now, so that material that's in this cell some of it could go across into this cell. Does that make sense? Got any questions on any of that? Because we're almost out of time, or essentially out of time. Okay, and then so I did make a little summary to this whole video, the whole chapter, which is cells are made of lots of parts integrated together. Forming a whole that's greater than the sum of its parts. You put them all together, it's alive. Right? Any one part of that cell by itself wouldn't necessarily be alive. You put them all together, and now you're cooking there, right? Which is interesting because at what point do cells become alive, right? If you take a part of it out, it could still function for a while, but it can't have all the function of the cell. It can't. Put them all together, and you got things that are alive. But if you could damage your cells, like I talked about slamming my fist down or whatever, that would kill some cells there. But look at that, they're going to be repaired. So you might think about how much can I damage a given cell and mess up some of its parts before it would not be able to recover and maybe not be alive. All right, that's all I got for you today. Hope you. I will see you tomorrow. See some of you today from 3 to 5, and others of you tomorrow, and all of you tomorrow at some point.
Wait, the last item.
Yeah. Today, I've got a class in there right up until 3, so I'll just stay in there. Oh, what do you think? This is cell. This is the completed cell. Yeah. Yep. I'm sorry, but I didn't get. Oh, let me do one thing here first.